Welcome to Caltech. My name is Kitty Cahalan, and I'm the Assistant Director of Educational Outreach at Caltech's Center for Teaching, Learning, and Outreach. CTLO is dedicated to building Caltech connections with students in Pasadena, Greater Los Angeles, and across the country. Alongside colleagues in Caltech public events, we work to ignite curiosity, mentor future scientists, and offer you all a glimpse into the exciting science and engineering that happens on our campus. We have a teaching program at a local elementary school, attend science nights at schools near Caltech, create summer research opportunities for high schoolers, and we help identify great speakers for the Science Journeys program. That's why we're all here today. Caltech has been presenting science talks like this one for more than 30 years. The science journey you're about to be part of will weave together cutting-edge research with the life story of one of our up-and-coming scientists. We're happy to have you here as our live audience. Our speaker today, Jess Kanwal, is a postdoctoral scholar in the laboratory of Professor Joe Parker. Jess grew up in Northern Virginia and attended the University of Virginia, where she majored in biomedical engineering. She earned her bachelor's degree in 2013 after conducting research in the laboratories of Dr. George Gillies, along with several other research projects you'll be hearing about today. She undertook her doctoral studies at Harvard University, where she worked under Drs. Aravi Samuel and Ben de Biver, earning her PhD in neuroscience in 2019. We're very excited to have you here today to hear about her journey. Welcome to the stage, Jess.
So nice to see all of you here today. I'm really excited to share with you some of the work that I've been doing here at Caltech. And I'm really excited to take you on a journey inside the insect world. Now, if you're like me, when I was your age, middle school, high school, you might think of insects as these annoying, maybe even irritating creatures that you don't often think very much about. But today, what I hope is that by the end of this talk, you'll have a new appreciation for insects and how we can actually study their nervous systems in order to understand basic brain functions and how organisms interact with each other in the natural environment. So some of you may have been looking at this video for a while now, and you might be wondering, what is this actually showing? Well, here you're seeing 4 different rove beetles, which I'll tell you more about in a minute, as well as their natural predator, an ant. And at first, it might look like the movements and the interactions that are happening are chaotic and random, but the more and more you watch these and stare at these interactions, the more you see that each behavior that the animal is performing is precisely timed, it's well coordinated, and it's responding to feedback from the other organism. And so for someone like me who watches these behavior videos all the time, all day long, it starts to look like a dance between 2 different species. And I hope that by the end of today's talk, you'll start to realize just how precise each action that these organisms are performing actually is. I want to step back for a moment and tell you a little bit about why beetles are an interesting organism to even look at to begin with. And that starts by first thinking about what class where beetles belong to, which are insects. Insects actually make up more than 70% of the species on our planet. And so as you can see here, insects which are shown in this pie chart, represented by the yellow, are actually the majority of organisms that live on our planet. And we as humans belong to this really small sliver right here that's labeled as chordates, and that includes all vertebrates, including mammals, fish, us humans, and any other species with a backbone. But as you can see, we actually are only a very small percentage of the different species that exist here. And so what we still have to understand is how this wide variety of insect species, there's likely over 1 million different species, many of which have not even been discovered yet, that exist, and how are they actually evolving and interacting and sensing their world. And when we look a little bit more in detail at insects and all the different types of insects, we can see here that the insects are broken down into different categories. And if you look, we find that, again, in this pie chart, we see that there's one category that's very predominant, and that's beetles. And so they're part of this order called Coleoptera. And beetles, as you can see from this image, really come in a huge variety of different shapes and colors and sizes. And so they have evolved to live in many different environments. Now, in particular, I'm going to tell you about a specific family of rove beetles called rove beetles, and those are shown here. So you might be familiar with some of the beetle species that are shown here, or families, like the ladybug, which has a large hard shell. But the rove beetles are actually ones that you may not have really encountered very often. They have a very different body morphology or shape to them. And what basically characterizes these beetles is that they have this flexible abdomen, which I'll talk a lot more about in a moment. But what's also really amazing is that these beetles have evolved to live in all different types of environments on Earth. So that includes the forest floor, inside of caves, under bark, invertebrate nests, even invertebrate dung. You can even find them in water and intertidal regions. So something about these beetles, which are one of the largest families of beetles, has allowed them to really adapt to so many different environments. And what is it that makes them so special for this? Well, one of the reasons I already mentioned has to do with their body shape. And in particular, these beetles, unlike some of the other beetles that you might have seen, they have what's called an elytra, which is this encasing right here. It's a hard encasing, and it protects the wings. So the wings fold up, and they are underneath this encasing. And most beetles have a pretty large, hard protective covering, like in this type of lady beetle. But the rove beetles have actually shortened this region called the elytra. And instead, they have exposed their abdomen, which is very flexible, and this allows them to very flexibly move through different environments. And so it's thought that these two components, these two morphological features are partly what enabled them to really diversify and live in different places. Now, in addition to this, they have also evolved different types of chemical glands, as I'll talk about in a moment, to really enable them to interact with different organisms. So as a rove beetle, what's your major predator? What's going to be the thing that you have to really worry about and try to survive against? For many animals, including these beetles, that live especially in forest floors, they are having to interact with ants. And if you've ever seen ant colonies, these are massive, often comprised of hundreds, if not thousands, of different organisms. And so this is just the very entrance of an ant colony, but you can imagine that just beyond this tree, there's a whole trail of additional ants and many other trees with additional colonies. And so it's well known that these ants are quite vicious. Whether it's this species in particular or many others, because they form this big colony with a lot of organisms, anything that is not part of their colony often can get killed or attacked. And many of these ants have these really sharp mandibles, which are shown here, that allow them to really crunch down and kill anything that they recognize as an intruder. So if you want to be this close to an ant, you have to really have some tricks up your sleeve on how to survive. And that's partly what these beetles have evolved to do. So this is an example of a different rove beetle, and you can see just how close it's managed to get to the ant and without actually being attacked. So what is it that has allowed the beetles to survive in these really hostile types of environments? Well, I'm going to show you 3 videos. These are going to be 3 different rove beetle species. And what you're going to see is the different methods and the different ways in which these beetles have evolved to live, coexist with the ants. And so as you're watching these videos, I want you to think about what you're seeing and how is the beetle actually surviving? What is it doing, and what behaviors are allowing it to exist with the ants? So here's the first one. This is going to be a video of the rove beetle Dalodia coriaria, which I'm going to talk a lot more about for the rest of the talk. But here you can see what happens when an ant comes and tries to chomp down on it. And here's another example, a little bit higher resolution. You can see that same beetle coming up towards an ant, which is flaring its mandibles. Now here's another example. Here you see this is a different rove beetle, and it's much bigger. But as you can see, it is approached by several ants. And you can observe certain behaviors happening here. Here's another one of these species. And so just think about what might actually be happening, and how can we just look at these behaviors in order to figure that out. And finally, here's another rove beetle shown here. And you can see it has a really different behavior from the ones that I just showed previously. So what's actually going on? How is it that these beetles are able to survive with the ants? Well, as I kind of alluded to in the title, they have these different movements or behaviors or methods for trying to coexist with the ants. In this first one, what's happening is that this beetle is actually using a chemical defense system. So these beetles have evolved a gland that allows them to attack ant predators so that they don't actually get consumed by the ant. And so when they flex their abdomen, if you notice that beetle was flexing as soon as the ant tried to grab onto it, then you'll see that what happens is it starts to excrete a chemical called benzoquinone, which is toxic and irritating to the ants. And this chemical, when it's smeared or slightly sprayed onto the ant, is what resulted in the ant immediately letting go and allowing the beetle to then escape unharmed. Now, on the other hand, these other 2 species have evolved different mechanisms. So this beetle up here called Platyusa, the one that seemed to be allowing the ant to feed from it, it actually evolved a different chemical that it secretes. And this chemical is kind of sweet. We don't know exactly what it is, but it's something that calms the ants down. And so the ants come, they feed on it, and then they don't end up attacking the beetle. And so this allows the beetle to actually live near the ant colony, where it can then find its own food. And finally, in this situation, we have a very different behavior happening, where the beetle literally had no fear of the ant, but actually climbed onto it. And from this situation and work in our lab, what we found is that what's happening is this beetle is chemically disguising itself. And what it's doing that by climbing onto that ant and literally extracting or removing the chemicals of the ant and rubbing it onto itself. And this way, the ant is unable to detect the beetle and distinguish it from other ants in the colony. And that's because ants don't really use vision the way that you and I do. They really rely on chemicals and smells in order to recognize their nest mates versus intruders. And so this is another type of strategy in which the beetle becomes what we call a myrmecophile, or an ant-loving organism. And then it lives in the ant colony, and there it actually acts as a parasite, where it can feed on the ant larvae. And so these 3 different strategies are different ways in which the beetle has evolved to live inside or to live amongst the ants that would normally try to predate it. So if we think about these beetles, then they're not obviously only interacting with the ants that I just talked about. Those are certainly some of the most dangerous organisms that they're going to encounter, but these beetles are going to encounter a whole variety of different species. Like I mentioned, they are found in so many different environments, and so they could encounter certainly different species of ants, but also they have to find their own food. A lot of them are hunters, and so they have to go crawl through the forest, and oftentimes that means finding larvae of various other insects like fruit flies or other fungus gnats that are flying in the environment and trying to hunt them down. And so how do they actually distinguish what is a real threat versus what's not? And how do they make that decision of when to actually deploy a defense mechanism, like I showed you, versus just keep going about doing their normal, activities, searching for food, resting, and so on? And so what I'm really interested in is trying to understand how the beetle's nervous system is able to recognize other species, detect what's a threat, and then coordinate these defensive set of behaviors. And so in particular, from now on, I'm going to tell you about that first rove beetle I mentioned, Dilodia coriaria. It's the rove beetle that has a chemical defense system and flexes its abdomen in order to avoid predation. So just to get you into the mindset of thinking about how the nervous system works, because a lot of how we study behavior and answer questions like the one that I had just posed is we have to first think about what do we think the function of the brain is. So I'm going to ask all of you a question now, and that is, what is the primary function of the brain? There's many things that our brains do, and across different animals, it varies quite a lot. But I want you to just think for a moment, what do you think is one of the most important things that our brain has to do in order for survival? And I'm going to give you a few options, and then by show of hands, we'll see what everyone thinks. So is the primary function of the brain to sense the world through different senses, whether it's chemical sensing or vision or sounds? Is it to make decisions and decide where to go or when to eat? Is it for learning and memory so that we can learn and acquire new information and then access and retrieve that in the future? Is it to coordinate movements so that we can navigate the different environments and species that we interact with? Or is it feeling emotions so that we can express and use those emotions in our daily interactions? So take a moment, and I'm going to go through each letter, and if you could just raise your hand for what you think is the most primary function of the brain. All right. So how many people say A, sensing the world? All right. Okay. How about B, decision making? Okay. All right. And then C, learning and memory. Oh, okay, we got a lot there. And D, coordinating movement. All right, handful. And finally, E, feeling emotions. All right. Okay, well, from my end, I could see everyone in the audience, and it looks like we have C as our winner in terms of majority. But I'm actually going to tell you it's something different. And first of all, I'm going to say that all of these are for sure functions of the brain. And it's not too surprising that many of you would think learning and memory, because you as students are probably really focused on doing that right now, going to school, learning new things, constantly having to retrieve new information. But I would argue that one of the most important functions of the brain across the animal kingdom is actually for coordinating movement. So why do I say this? Well, if you think about it, a lot of those other functions are really in service of helping animals figure out when and how to move. And so here's just an example of how information travels and is processed through the brain. So imagine, for example, we have our beetle, and it comes across a variety of different species. Now, certainly, the first thing that it needs to do is actually sense the world. And how does it do that? It takes information about whatever organism it's interacting with. It might extract information about what it looks like, how it's moving, what it smells like, how it feels like. And so it's sensing the world. But those sensations are really there to help it decide how to respond to that other organism. Now, once that information gets processed into and starts to get extracted and filtered into the brain, then the animal has to make decisions about whether it knows from previous experiences and memories that that's something dangerous, or whether it needs to think about how its internal state, like whether it's hungry or whether there's fear from other potential threatening situations, and use all of that together to make a decision about how to respond to the animal. And so at the end of the day, all of these components are really helping the animal decide and get motivated on how to move. And so that's why I say that movement is one of the most critical functions that our brains, but also brains of all other organisms down to tiny little insects, performs. So what actually drives these movements? I kind of already alluded to that, but there's many different types of interactions that animals perform with each other that help promote the way in which an animal decides to move. So this could be really important predator-prey interactions. This is across the animal kingdom, so you've probably seen just how fast and agile certain animals can be in order to escape from predation. But it can also include interactions that have to do with allowing animals to mate and court each other. So here you can see a male bird dancing to try and get the attention of a female. Now there's also all sorts of competitive interactions between species. So these types of interactions you might even see in your backyard or in your playgrounds. You can see here that there is competition even between different organisms for the limited resource of a little seed. Or of course, a raccoon trying to get a little bit of food from our pets. And then finally, there's interactions like these, which is a host symbiotic interaction. And these can be mutualistic, they can be parasitic, but basically it occurs when different organisms end up relying on each other in some way. And so here you can see through this mutualistic interaction that this little cleaner fish is benefiting by being able to feed off of the teeth and the mouth of another fish without having to worry about actually being eaten by that bigger fish. And for that bigger fish, it's getting kind of like a free dental cleaning. So basically, these types of interactions are really critical to shaping how the brain functions and shaping, ultimately, how animals move in their natural environment. And so that's why it's super important to understand the brain with this idea in mind. And so coming back to this question that I posed specifically about the rove beetles and trying to understand how do they actually recognize other species and how do they coordinate behavior, the first thing that we really have to do to answer this question is to look at their behavior. And so we do this in really high resolution. Now, first I'm going to show you what happens if you just put a beetle and an ant together. This is a beetle and an ant in an arena in the laboratory. It's been sped up, so it's not actually this fast, but you can see it's really dynamic and complex and very, very hard to understand what's happening. There's so many different interactions happening. You might see a little bit of that abdomen flexing that I showed you earlier, but there's so much chasing and biting, and so it becomes quite challenging to really piece apart the different behaviors that are important for this interaction. So instead, one of the key ways that we can study behavior in the lab is by basically zooming in on this interaction and placing a bunch of different cameras all around the beetle, and at the same time, tethering the beetle a bit so that it can't just like freely run around, but put it on in such a situation that it feels like it can still walk. And so we do that by actually tethering the beetle on a ball, a styrofoam ball that's floating on a very thin layer of air. And this gives the beetle the perception that it's like walking on a treadmill. So it can keep walking, it can turn from side to side, but at the same time, it's fixed in one position. And then we can present an ant, just like an ant might come in the natural environment, and try to understand how exactly the beetle's body and movements change during this type of interaction. And so, as I was just describing, what we do is we basically deliver an ant, which is also tethered, but is alive, to the beetle at closer and closer steps. And then we can, again, really zoom in and see what's happening to the beetle's body and behaviors. So I'm going to show you a video of what that looks like. You'll see the ant approaching, and the little colored dots are the way that we track the animal movement and body postures. This is something that we do after we record the experiments using machine vision. But what you'll see is that basically as soon as the beetle makes contact with the ant, you can already see that it performs that flexing behavior. And so this is something that we can say is able to be reproduced in a laboratory setting. But even more interestingly, when we start to look at this behavior in more detail, we can really break down when and how the behavior is being performed. Now, I'll just quickly point out that this behavior, that really intense flexing that you saw, which is correlated to the release of those toxic chemicals, it doesn't happen with every insect that this beetle interacts with. So it should only happen with predators, and so to make sure of that, we also have tested other species like a fruit fly larva, and here this is an organism that the beetle would actually want to feed on. And so here you can see this larva, it's been tethered, but it is still alive. And you can see that as the beetle makes contact, it doesn't have that strong level of flexion that we saw towards the ant. And so, of course, depending on different contexts, this behavior can vary, but if the beetle was really, really hungry, it might try to actually feed on that larva in certain situations. And so by looking at the interaction between the organisms in this way, we have been able to see that there's two major behaviors that the beetle performs when it comes across an ant. There's a fleeing behavior, which is really hard to see from just those videos, but from additional analysis, really looking at the pose of the animal, what we find is that as the ant gets closer and closer to the beetle starts to turn away. And so before it even makes any contact, it's already sensing that there is some kind of threat, and it's trying to get away from the ant. Now, I forgot to mention that all these experiments are done in the dark, so beetles don't actually seem to use vision very much like we do, but they really rely, like many other insects, on chemicals. And so we predicted from these experiments that this response was likely due to some kind of chemical sensing that they're able to perform, whether it's smell or some other chemical cues in their environment. And then once they make contact with the beetle, they start to perform that flexing. And that behavior is really obvious, and you can see it from the videos. But basically, they don't do very much of that flexing in the beginning, but when they make that contact and have touch, they're able to really strongly elicit that flex and try to defend themselves. And so what happens now when you take away their ability to smell? So one of the things that we can do to really start to understand how the nervous system works in these insects is that once we understand the behavior, we can try to manipulate their nervous system and then see how does that affect the behavior that we've described. And so using genetic tools, we can actually go in and we can turn off their ability to smell. And the way that we do this is that these beetles have olfactory receptor neurons, which are their smell sensors. They're very much like the smell sensors that you and I have in our nose. And inside of these, these are a set of type of neurons. And inside of these neurons, there are specific proteins called olfactory receptors. And these receptors all have a common subcomponent called orco. And so we can actually go in and genetically modify this protein and basically cut it out of the genome so that the animal does not produce a functional olfactory receptor neuron. And this way, we can actually see what happens when you take away their ability to smell. So take a moment and guess, what do you think would happen if they can't smell? Yes, you want to guess?
If they can't smell, they can't see where they're going or nothing.
Yeah, that's a great response. So because beetles use smell as a way to see their world, a great prediction would be that they can't really tell where they're going. And in particular, we thought that this means that they also wouldn't really be able to tell whether they're interacting with an ant or some other organism, and that they might not perform that flexing behavior as strongly as we saw in the videos that I just showed. I'm not going to show you all the results, but what we end up finding is that they actually still are able to detect the ants. So even though they've lost a huge part of their sensory world, they have ways for still detecting something as threatening and dangerous as an ant. And a lot of this work is done in collaboration with other folks in the lab that I work in. So in particular, I just wanted to mention these two people, Mina and David, who helped create the genetic tools that allow us to manipulate beetle nervous systems. And so essentially what we find is that just like in us humans and just like in many other organisms, beetles rely on multiple different sensory cues. So even though they are no longer able to smell the ant, they still are able to respond and detect to the ant by flexing. Now what we find though is that each of these sensory modalities has a different weight or a different contribution to the beetle's decision of how to respond. So yes, they still respond to the ant, but they don't respond as well as as a beetle that has its full ability to smell. And so what we found is that smell plays one of the largest roles in making that decision, but even when it's gone, they can still use information about touch, or what we call mechanosensation, to make a decision on whether something is a threat. And so what a lot of this work has showed us is that the nervous system of even these tiny little insects with a very, very small brain is still robust to recognizing very threatening situations in other organisms by using multiple sensory modalities, just like you and I would use. So even if we end up having some kind of impairment, oftentimes when we grow older, we start to lose our ability to hear or maybe even smell, but we can still do a lot of the things that we used to. Maybe not as well, but we can still do them because we have other senses that are compensating and helping us to do those behaviors. And the beetle is very similar. And so that's also where redundancies get built in that allow the nervous system to still allow the animal to perform really important behaviors. Okay, so what's next? How do we actually understand how the brain is encoding an ant versus something else? Well, ideally, we'd actually like to look inside of the brain and see how cells in the brain are responding when a beetle interacts with an ant. And that's something that our lab is still working on, developing tools so that we can do this. But I want to show you just some of the very first steps we are taking to look inside of the beetle brain. So this is a beetle nervous system, particularly the same beetle that I talked about called Dolodia. It's got a brain up here in the head, and then just like you and I have a spinal cord, it has a ventral nerve cord that comprises of smaller subsets of neurons that control a lot of the legs and a lot of the body parts. And we can go in and start to actually use fluorescent proteins to start to map out the nervous system of these beetles. So here is a stack from a confocal microscope where we're basically zooming through the brain of a beetle. And what you can see is that there's different structures here. You can see certain kind of patterns in the shades of gray And as you get to different slices of the brain, you start to see different structures form. And we are just starting to understand what these our structures are. So we've created a map of the beetle brain, and thanks to a lot of similarity between this beetle's brain and many other insect brains, we can start to identify areas of its brain where it uses smell, where it processes smell or taste, or any combination of these. And ultimately, what we'd like to do in the future is then go in and actually record from neurons in these brain regions and see how they're responding to an ant and how they're able to detect something as threatening as an ant versus some other species the beetle might encounter. So that's a little bit about the research that I've been working on here in the Parker Lab with beetles. But before I keep going, I want to just step back and tell you a little bit about my journey and how movement has played a role in that journey as well. So I'm going to tell you a little bit about the different types of movements that have popped up along my science career. So it all kind of started for me as a little kid. I really enjoyed dance, and my parents are from India, which is a culture, they come from a culture that is very, very much surrounded by dance and music and celebration. And so as a kid, that's something that I just naturally loved doing. A lot of the movies and a lot of the festivities in this culture really revolve around dance. As I grew older, I started a little bit along this science journey because I was introduced to a set of books by a close friend that were science fiction books about astronomy, about the world beyond Earth, and I became super fascinated with what happens beyond our planet. And so I got really into science fiction books like 2001: A Space Odyssey and Contact, and for a long time, I really thought that I was gonna go study astronomy and learn about what happens outside of Earth. But then a few things happened that kind of changed that path when I was in high school. So the first is that my grandma, who's shown here with my brother and I, she moved close by to us, and she also developed Parkinson's disease. And this is a neurodegenerative disorder in which there starts to be deficits in movement. And so over time, I would start to see how her movements started to change. She would start to have trembling hands, sometimes not even be able to easily pick up food as she was eating. And at the same time, I also started learning dance a little bit more formally. And so I started doing Bhangra, which is a type of Indian dance from Punjab, which is the region of India that my parents are from. And I started learning a lot more about movement in that sense, in terms of how my body was moving, and how to learn choreography. And so these two things, I think, both kind of propelled me to wonder what actually is happening in the brain to either allow for our bodies to perform new movements or to actually take away our ability to perform smooth, flexible movements like we have for much of our lifetime. And around that time, I also had the first opportunity to actually look at neurons under a microscope. And that was just so fascinating for me. It was this beautiful fluorescent green glowing set of neurons that were all connected to each other. And I remember going into a research lab, and the advisor, the professor there, took me into this dark microscope room, and she had a slide with all these neurons that had been basically plated on the slide, and it was just amazing. It was like all these different cells connected together, and somehow those cells were allowing us to perform all these different functions, whether it was movement or learning and memory or any of the other things that our brain does. And so I got really interested in, instead of looking at what happens beyond our planet, what happens within our bodies, and specifically in our brains. And then from there, I went kind of on a science journey in various different labs. Now, originally, I was really interested in trying to work in research that might be related to understanding Parkinson's or movement, but at the end of the day, I never actually ended up working in a lab that did either of those things. Instead, I kind of just took whatever opportunities for research labs existed and seemed interesting. And so that took me around to actually various different parts of the world to work in different organisms, whether it was fruit flies or honeybees or fruit fly larvae. And as I worked more and more in these different insects, I started to become fascinated by how they behave, how they survive, and how they sense the world. And I even was really excited to learn that some of these insects and many other organisms kind of perform their own dances. So if you've ever seen honeybees in their hive, they perform what's called a waggle dance. And this is basically this buzzing movement, this vibrational movement that they perform, and it actually is how they communicate to other hive nestmates about where food is and how far away it is, what direction it is from the hive, and how good of a quality that food is. And so I just thought it was so amazing that a tiny little insect like a honeybee could perform such a complicated yet amazing form of communication and behavior through dance. And so eventually I ended up doing grad school where I studied how fruit fly larvae, which are much more behaviorally simple, but still very smart organisms in the sense that they have to find food so that they can grow and develop enough and become adults that look very different from how they are initially. And how do they use different senses, whether it's smell or taste, to find food and the most nutritious part of their environment? And then from there, I got really interested in where I am now, trying to understand rove beetles. And I heard about this work here happening at Caltech, and the beetles sounded super interesting. And so I wanted to come and really understand what's happening in their brains, because very few people have even studied the behavior of these organisms. Now, all while doing research in these different places, I continued to also find ways in which I could continue to learn dance, specifically Bhangra, and change and increase my own movement in that sense. But for a long time, these two different interests of mine were quite separate. And it's only recently, like around 2020, literally during the pandemic, that these things started to come together for me. So during the pandemic, I had just joined the lab just a few weeks before everything shut down. And as a result, what ended up happening was that because I didn't really have any data to work with, I ended up bringing a little bit of the lab into my apartment. And what that meant was basically bringing this little behavior setup and some beetles home and setting it up on the dining table of our apartment and just watching how these beetles behave and how they interact with other species. I didn't really know much about the beetles yet, and so it was a great opportunity to just become familiar with their movements and the ways that they move differently when they interact with different species. So I'd go out during the day, collect some insects, bring them back, and then place them with the beetles and see what happens. And you could see all these really interesting behaviors happening. Sometimes they would ignore the beetles or the other species. Sometimes they would try to chase them. Sometimes they would do these interesting spinning behaviors that looked like some form of dance. And so it was just a way in which I really started to appreciate the different movements of these organisms. Now, at the same time, I also wanted to continue dancing. Of course, that couldn't happen in person, so I started doing virtual classes. And basically, the living room became my dance studio. And so these two spaces that were very close to each other basically now started to intertwine, where I was doing science and dance in the same space. But in addition to this literal physical proximity of these two interests, I also started collaborating with other people and really trying to think about the intersection between science and dance. And what that resulted in was a publication or a review paper in which I reached out to a dance professor to think about how they actually study movement and behavior. So, like I showed you as a scientist, I had been looking at animal behavior and thinking about how to break it down, whether it was the beetles flexing or fruit fly larvae traveling through its environment. How do we study behavior in order to understand the brain? And in that process of thinking about how best to do that, I had come across in the dance world a technique that the dancers use to annotate dance movements or dance sequences. And it turns out there's a lot of similarities between that type of interaction or that type of analysis in the dance world and in the science world. And so we collaborated to try and understand what are the things we can learn from studying movement and behavior as a dancer versus as a scientist. And so don't worry about all of the complexity shown here, but I just want to show you as an example, this is actually the way in which dancers annotate movement. And basically the way this reads is kind of like a music staff, except from left to right, it goes from top to bottom. And the different symbols represent different different parts of the body. And so here's an example where we basically took a video of a Bhangra dancer named Gurdeep. And this is him dancing right after getting the first COVID vaccine, so he's really happy. And we basically, for the first time, were able to annotate what that looks like. And you can see at each of these points, this is the frame, and these are the hand movements, the leg movements that he is performing at that moment. And so what's so cool is that just from this one diagram, someone who understands how to read this particular stuff can then perform this behavior. And that's a lot like what we do as scientists when we're trying to annotate how animals are performing and behaving. And we try to use symbols similarly to represent those movements in different parts of their body. So this really brought together for the first time for me both these interests in science and dance in a way that I had never really expected or anticipated, and really made me think about the ways in which we can learn from these different fields to help improve the other. And so now I do a lot more of this type of stuff. And I've been able to really work with dancers of all different types. So I've told you a lot about Bhangra, which is this very energetic, joyous dance. But working in the LA area is so nice because there are so many people with different dance expertise, from West African dance to Venezuelan dances. And so we have been able to work together to try and share what it's like to dance, and then how our brains are coordinating that, and how dance in turn coordinates and affects our brain. Now, I just want to end with a few just key lessons from thinking about the intersections of science and dance, or science and arts in general. So one of the things for me in particular that became very obvious was that by thinking about science and dance, I realized that there's a lot of skills that overlap in both. And one in particular was really this new skill of being able to take very detailed observation of body posture and movement sequences in order to understand how something is working. And so like I already showed you, with the beetles, a lot of what I did was watching these movies, these videos of beetles interacting with other organisms, and breaking down that complex behavior into these two parts, the fleeing and the flexing, and then using that to understand how the nervous system controls these different behaviors, which we call behavioral motifs. And when you think about dance, dance is actually very similar. So when we learn dance, we often will learn smaller movements that then get put together in different ways to create a dance sequence. So here are just a few different movements of me performing individual little little movements in Bhangra in particular. And you can see that there's like 6 different moves that I've showed here. And you can put these together in different combinations in order to get something more complex that is then coordinated to the beat of music or to however you're feeling. And you can put these together in different ways, and you get different types of movement sequences or different dances. And so there's a lot of similarity then in how these little insects perform. They can put together the flexing and the fleeing in different ways depending on the context, and also perform different movement sequences to escape from predation depending on the environment and the situation. Now, there's a lot more things that I found as I thought more and more about the connections between science and dance. I won't go through all of them, but just to say that there's so many aspects and skills that I think we can learn from other aspects of our life that can influence how we perform, whether it's science or how we do the arts. And so just to give you a few more examples, certainly recognizing patterns is a huge part of science. We have these big data sets. We're constantly trying to find patterns in them so that we can understand how animals function. And that's actually a huge part of dance too, because as I just showed, when we're learning new sequences, often we are trying to look for those key movements or patterns that might reappear over and over again and understand how and when we can incorporate them into a new choreography. There's also a lot of emphasis in both science and dance on repetition. Obviously, as a dancer, you perform a dance many times before, you practice a dance many times before performing it. And a lot of times this involves a lot of trial and error. It requires a lot of resilience. And a lot of that is very similar to how we do science. Oftentimes, a lot of our experiments fail at first, but we try to learn from those failures and then repeat and do it again. And a lot of both science and dance involve collaboration. Especially if you're doing a group dance like Bhangra that I showed, you have to coordinate with the other people in the group. And similarly in science, a lot of the work that we do is really a result of teamwork and collaborating with other people that come with different experiences and different ideas. So there's a lot of interconnectedness between science and dance, and I would argue between any of the arts and sciences as well. Just to give you a few more examples, this is Santiago Ramon y Cajal. He's considered one of the fathers of neuroscience. And he was not just an amazing scientist, but he was actually an amazing artist as well. So these are drawings that he made. And actually these drawings have led to some of the key core concepts and our understanding of how neurons are connected and how they develop and become the nervous system. And another example is Mae Jemison. She is the first woman of color to be an astronaut. And she's also an amazing dancer. And if you're interested, she has some great videos of how dance informed her experiences as an astronaut and vice versa. And so if you're interested at all in the intersection between these different fields, you can also look to some of the previous Science Journey talks that have happened here at Caltech, where there are several amazing graduate students who talk about the intersection of their type of artistry, whether it's drawing, animation, or music, and how that's impacted and paralleled the skills that they have developed as scientists, and vice versa. So I hope today you have learned a little bit more about how organisms interact in the environment, how by studying their movements and behaviors, we can understand more about their nervous system, and how social interactions are super important for the types of behaviors that we see animals perform. And I'd just like to end by asking you all to think about what moves you, what are the things that you're excited and passionate about, and how do you think you can choreograph your journey so that you can integrate them in unique and different ways that may not necessarily mean just picking one over the other. So with that, I'm happy to take any questions, but I first want to give a quick shout out to the amazing people that have been involved in some of the work that I described, all the organizers here that made the science journeys possible, especially Mary, and really this opportunity for all of us to come together and talk about science is super fun and awesome. And with that, I am happy to take any questions you guys have. Thank you.
All right. Thank you. In the interest of time, since we got started a little bit late, we're actually just going to take 3 questions. So, first up to the mic gets to go.
What's your favorite type of bug?
That's a great question. My favorite type of bug is actually the honeybee because of that cool waggle dance that I showed you. I think it's just fascinating that it's able to, purely through movement, it can't speak like you and I, but it's able to communicate really complex information, like how far away food is or how good that food is, just by the way that it waggles and moves with the other bees in the colony.
How did you tether the insects?
Yeah, that's a great question. So it's really challenging. You have to make sure that your hands aren't too shaky. But essentially what we do is we can place the beetles or the ants on a very cold surface. And that kind of knocks them out for a little bit. And so they don't move around too much. And then we take a special kind of glue that is basically liquid. And when you shine a certain wavelength of light, UV light, on this glue, it hardens. So we take a little dab of that glue, we put it on the beetle, and then we shine the light, it hardens, and then the beetle is just very gently stuck onto that little piece of wire. And we place it on its back where it won't really disrupt its ability to walk and move around.
One more question? You want to come up to the mic?
I can repeat your question if you want to shout it out. Okay, the question is, have I ever gotten bit by a bug? Yes, for sure. And I think that's one of the reasons why when I was younger, I didn't really think much of insects. I didn't really find them interesting. I was really, really one of those people that mosquitoes find super attractive. And so I got bit by mosquitoes quite a lot, and also definitely got a few bee stings, so yeah. No, so the beetles don't, these beetles at least don't bite us, and they don't really bite anything except for very soft-bodied organisms like fruit fly larvae. So that's really nice. But I've definitely gotten bitten by the ants.
Awesome. Thank you so much. One more round of applause for Jess.